Today's presentation is, what does the Clin Pharm plan for Biologic look like? Our speaker today is Dr. Khalid Ben-Kali. Khalid is currently a director of clinical pharmacology at Certara. He's a pharmacist by training and also has earned Master's of Science degrees and a doctorate in pharmacology, as well as an executive MBA. Before coming to Certara, Khalid spent 10 years in the pharmaceutical industry at Galderma and Pierre Fabre, where he participated actively in the development of new chemical entities and monoclonal antibodies. Welcome to the presentation, Khalid. The floor is now yours.
Thank you very much, and thanks all for attending this webinar entitled What Does the Clin Pharm Plan for Biologic Look Like? So as an introduction, the biologics, for most of them, the developed and the approved one, are monoclonal antibodies. So I will focus my presentation on monoclonal antibodies. The majority of these monoclonal antibodies or MABs are derived from IgG1, and this share the characteristic of this subclass of antibody. This class of antibody consists of two fragments, the fragment antigen binding or Fab, and this region mediates the interactions with the target antigen. And the second fragment is the fragment crystallizable or FC, and this region interacts with a variety of accessory molecules to mediate indirect effector functions, such as the antibody-dependent cellular cytotoxicity, IDCC, antibody-dependent cellular phagocytosis, IDCP, or the complement-dependent cytotoxicity, CDC. For many drug development programs, it is possible to leverage the prior knowledge about the monoclonal antibodies because they are sharing the same characteristic. So the objective of this webinar is to help you to not reinvent the wheel each time and to help you in your project by gaining and using the knowledge already available for the development of this class of therapeutics. In the first part, I will present the ADME characteristics and the variability factors related to the pharmacokinetics of the monoclonal antibodies. So for the absorption, the administration route of choice for the monoclonal antibodies are either intravenous or subcutaneous route, and sometimes we can use the intramuscular one. For obvious reason, the oral route cannot be considered for these compounds, otherwise they will be degraded in the gastrointestinal tract. After subcutaneous or intramuscular administration, they will enter the circulation presumably through a lymphatic fluid drainage, and they will exhibit a typical biphasic PK profile with a relatively fast distribution phase followed by slower elimination phase. This results in a half-life ranging between 11 to 13 days in human. Also, following subcutaneous injections, the maximum plasma concentrations are often observed around 3 to 7 days, and the bioavailability varies between 50 to 90%. For the distribution at steady state, the MABs generally exhibit a low volume of distribution, ranging between 3 to 8 liters, and this is representing the confined distribution to the vascular and the interstitial spaces, and this is because of their size. They are relatively large molecules and their polarity. The primary mechanism of antibody diffusion in tissues is through the convective transport through paracellular pores in vascular endothelial cell membranes. Now regarding the metabolism, or we can call it also the catabolism and the clearance mechanisms, the MABs are mainly metabolized and eliminated through proteolytic degradation. This will result in smaller peptides and also in amino acids. The clearance pathway of the use in the metabolism and the elimination could be classified in three mechanisms. The first one is the non-specific clearance, or what we call target independent. This will occur through a non-specific endocytosis in cells and proteolysis in liver and reticuloendothelial system. Generally, this non-specific clearance or target independent will result in a typical linear PK profile. The second mechanism, target-mediated specific clearance, this will occur through the antigen-specific interactions. So after that, the MAB binds to a specific antigen, which could be soluble or membrane bound, they may be internalized and catabolized through lysosomal degradation as an antibody-antigen complex. However, the target-mediated clearance decreases with the saturation of the target, and this will result in what we call target-mediated drug disposition, or TMDD. With this second mechanism, generally, we observe a nonlinear PK profile. However, it is important to note that both mechanisms, the target-mediated specific clearance and the non-specific clearance, can coexist for the same compound. However, this will be either predominant at low doses or high doses. So at low doses, we'll observe a nonlinear PK profile due to this target-mediated specific clearance. However, once saturated, the non-specific clearance will take over and we will observe a linear PK profile at high doses. The third mechanism related to the anti-drug antibodies, or ADA, and these are immune response and resulting from the formation of immune complex with the MABs. This clearance mechanism is not related to a specific compound or drug, but may be observed in some subject developing a neutralizing ADA. And these complexes are varying in size and structure, and they are cleared at a different rate than the monoclonal antibody alone. So we can see in the third curve that for some subjects, we may observe a rapid decrease of the concentrations, and this is due to the rapid clearance observed in those subjects developing neutralizing ADA. So as for other products, there is a high variability in monoclonal antibodies. The sources could be related to the product itself or related to the subject. So for the product-specific variability factors, we can denote the charge, which is one of the major determinants of how the mAb will interact with negatively charged component of the cell surface. So for example, during the monoclonal antibody engineering, the isoelectric point can be modified, and this modification will result in modification of the clearance, the distribution, and even the bioavailability. The glycosylation pattern is also an important source of variability and a critical one for the monoclonal antibody. So the modification of the glycosylation will result on an impact on the pharmacokinetics or the pharmacodynamic profile. There is one known example of a marketed product, tartuzumab or Herceptin, which has 8 different isoforms, and each isoform is related to a specific PK/PD profile. For the factors related to the patients, the most important one is the body weight. We saw previously that the distribution volume is limited to the plasma and extracellular fluids, and the value of the volume distribution is relatively low. So if you observe a limited variation in blood volume, this will result in a significant change in systemic exposure, and this may be translated by clinically meaningful differences between subjects. So to compensate for the effect of body weight, the monoclonal antibodies are administered generally either using a body size adjusted dosing on milligram per kilogram of body weight, or we can define what we call a weight-based target fixed doses, meaning that we would define relevant body weight ranges where the variability is acceptable in terms of pharmacokinetics and pharmacodynamic or response, and then we'll have a flat doses for each range of body weight. But generally, a fixed dose approach is preferred as it offers convenience, compliance, cost effectiveness, and also, most importantly, the safety, because you will have less risk of dosing error with a fixed dose than a body size adjusted dose. The other important variability factors related to the patients, the immunogenicity, we saw that the ADA may have an impact on the clearance, so this will be translated by difference in PK, but these differences in PK could be also translated by differences in efficacy and sometimes the difference in safety profiles. And what is complex with the ADA can be transient, can be persistent, and also the incidence can be impacted by several factors, which will increase the intersubject variability. The inflammatory status is also identified as a variability factor, and this is relevant for inflammatory diseases. So for some, depending on the severity of the disease or the degree of the severity of the disease, we may observe different proteolytic degradation, and this will be high for the most inflammatory diseases. So this will modify the clearance between subjects. The genetic variants, so the genetic polymorphism for genes encoding for protein relevant to the distribution of the elimination, such as the FcRn of the Fc gamma receptor, could be also translated by intersubject variability. The last one is the ectodomain shedding, and this phenomenon is encountered for membrane-standing receptor, in which the extracellular domain of the receptor may be cleaved and released to the circulation. So the circulating level of this receptor may vary between subject and will introduce an additional level of variability between subject. This was observed, for example, for almetizumab targeting the CD52. Now, in the second section of this presentation, I will focus on specific studies, clinical pharmacology studies for monoclonal antibodies. I will not go through the standard studies, but really on the studies which are relevant and specific to this category of drugs. So as you know, the monoclonal antibodies are complex compounds, and we may face a lot of manufacturing changes during the development. This is quite usual situation, and this manufacturing change may occur on the substance, on the formulation, and also on the primary container. And all these changes may trigger a modification of the biological profile, including the pharmacokinetics, the immunogenicity, the safety, and also the efficacy. However, what is important is to evaluate the risk related to these changes. So generally, the approach is more you are close to the registration of the product or you are conducting your pivotal trial, the risk could be higher for patients and specifically for safety. For this reason, the comparability expectation may be different depending on the stage of the development, where you could use, for example, only analytical characterization or non-clinical assessment for early stages. At the late stages of the development, clinical assessment is mandatory and will be critical and key for your development. One of the usual situation of the manufacturing change is the switch between vials from vials to prefilled syringe or autoinjector. So often with the development of biological drugs, we start using vials containing the drug. This is really convenient to test different doses. However, this will need intervention from trained personnel, healthcare professional, and this will introduce complexity and a high cost. So this is acceptable for early stage studies. However, when you are close to large scale studies or commercialization, you will need a more friendly devices to administer the drug. So generally, we switch to pre-filled syringe or auto-injector. However, this change may introduce additional variability impacting the bioavailability, the administered dose, or the precision of the administered dose may be different. The immunogenicity could be also different between these devices. And of course, we should also take into account the human factor. So there is a variability depending on who will inject the product. So there is a difference between old patient and the healthcare professional in terms of precision of administration. So generally, if we switch at the late stage of the development, we'll need clinical assessment. So a dedicated comparability clinical study is needed to investigate these differences. However, you can always consider alternative approaches. So depending on the discussion with health authorities, you can, for example, conduct population PK analysis if you have the data for example, a subject or a subset of subject or data with the different devices. You could also consider collecting data from what we call usability assessment in real world. Generally, you can have, for example, phase 4 studies where you are using these devices. So you could consider collecting additional information such as the PK and also the dose precision in these populations. In terms of design of the clinical PK comparability studies, we may prefer a simple studies, meaning a single dose in healthy volunteer. However, this is not always feasible and possible, and this will depend on the profile of your drug. For example, if you have any safety risk with your drug or you have a specific PK profile in your patients, so you cannot enroll only healthy volunteer. Patients are needed to investigate accurately the PK profile. Also, if you are targeting, for example, specific concentrations to achieve pharmacodynamic effects, or you would like to assess the immunogenicity in your comparability studies, you will need to administer drug in a repeated way, and then the single dose study will be not feasible in this case. In terms of sample size and the study design, generally we use standard bioequivalence criteria to evaluate the difference between pre-change and post-change in terms of manufacturing change process. However, these bioequivalence criteria are not a regulatory requirement. Nevertheless, in terms of calculation of the sample size, generally will lead to a high number of subjects, and this is not due to the bioequivalence criteria, but this is due to the parallel design. As we saw previously that the elimination is relatively slow of this drug, so you cannot consider washout between two sequences to compare this drug. So generally, you need to have this parallel design, and this will increase the number of subjects needed for the comparability. Always keep in mind that flexibility may be granted upon discussion with the health authorities, and this will concern what is the definition of clinically meaningful differences. So the difference in PK itself is not sufficient to define this, but sometimes you may negotiate to say that yes, we have some differences on the PK, but this is not considered as clinically meaningful. You have in this table some examples of recent comparability studies for several drugs. So you can see in this table that they enrolled either healthy volunteers or patients. Yeah, they use generally a single dose regimen. There is one example of repeated dosing, but for most of the cases that were using a parallel design, there is only one study where a crossover was used. When you see to the column of the sample size, you can see that there is some variability. So this will depend on the objective of the study, but generally we have relatively a large number of subjects. So here we are ranging from 7 subjects per arm to 175 per arm, so which is quite a significant number. However, what is interesting to see in this table, the two last columns, so there is a question, does the PK meet the bioequivalence criteria? So we can see here some example where the answer was no. However, when you look to the last column, where is the PK comparability accepted? So the answer is yes. So this is related to the clinical meaningful differences. So you could have some PK differences, but it is acceptable to bridge between before and after the change. The second aspect, which is really specific to the monoclonal antibodies, is the investigation of the DDI. So up to recently, we were quite in the dark, and the investigation of the DDI was relatively complex and difficult. However, now we have a new draft guidance from the FDA, which clarifies a lot how to investigate the DDI, the approach, and the different mechanism. We don't have EMEA guideline, but probably we'll have one soon. So in terms of DDI for monoclonal antibodies, you need always to evaluate the risk related to the DDI. So you need to consider the pharmacology and the mechanism of action, the clearance mechanism, and also the co-administered medication in patients' population. What is interesting in terms of DDI, so you have all the situations possible. You may have DDI occurring between large molecules and small molecules, between the biologics themselves, and also the biologic drug may be a victim or a perpetrator. In terms of DDI mechanism, in this guideline, they are classifying in two categories. The first category is related to the pro-inflammatory cytokine mechanism. And the second one, the second category is not a single mechanism, but they are involving all the mechanism except the one related to the cytokines. So in terms of pro-inflammatory cytokine related mechanism, this is known since now a significant time, and we know that pro-inflammatory cytokine level can downregulate the expression of the cytochromes, thereby decreasing the metabolism of drugs that are substrate of these CYPs. So we may observe an increase of the level of these drugs. And we know also that if you introduce a therapeutic which can modulate the pro-inflammatory cytokine, you will modify the regulation of the CYP expression. So you will also modify the activity of the CYP substrate. So here you have an example of this interaction, which was investigated for tocilizumab, an anti-IL-6 drug, which was investigated in rheumatoid arthritis population. In this population, they have a high level of anti-IL-6. So when they are using simvastatin, we can see here in this graphics at day 1 that they have a high level of simvastatin exposure. However, after repeated treatment with tocilizumab, so we have a modulation of the expression of the CYP, and you can see here that the simvastatin levels will decrease because we are restoring a normal expression of CYP3A4. So this is not an induction, not an inhibition, but we are modulating the expression of the CYP3A4. In terms of study design, how to investigate this DDI interactions, as you know, as we are modifying the CYP expression, so this will not concern only one isoform, but probably it will impact several isoforms of cytochromes. So you are obliged to conduct what we call a cocktail DDI study. So you need to administer a cocktail of probes to cover all the relevant CYPs. So here you have this example of risankizumab, anti-IL-23. So in terms of design, at day 1, the investigators administered a cocktail of the probes before starting the treatment with the biologic. They measured the concentrations and measured the PK parameters, and then they started the repeated treatment, here in this example for 13 weeks, to be sure that we are modulating the expression of these CYPs, if any. And then at the last day, we'll re-administer the cocktail of the probes and assess the PK of these probes. And you can see in terms of analysis here that in this forest plot, we are investigating the ratio between the after the treatment and before the treatment. And you can see in this example that there is no clinically meaningful DDI. For the second category of the DDI, so I said before, this is not a single mechanism, but we're grouping all the possible DDI mechanism. Here you have 3 examples. For example, the biologics affecting a physiological process. There's one example of dulcalutide, and this peptide may modify the gastric emptying. So there is a theoretical possibility that we may impact the absorption of the orally absorbed drugs. Second example of the co-treatment with immunosuppressants. So here we may impact the incidence of neutralizing ADA. So we saw before that the ADA may impact the clearance of the drug in some subject. So in the example here of the adalimumab, if you administer it with the methotrexate, you will notice that the clearance is modified in some subjects. So here they estimated between 29% and 44% in rheumatoid arthritis patients. Third example of the modification that they can impact the target or the target-mediated disposition. In this example, evolocumab, an anti-PCSK9. In this case, if you administer a drug which may modify the expression of the target, in this case the statins, you may notice also the modification of the clearance of the biologic drug, because its clearance is dependent on the expression of the target. So here they observe approximately a 20% increase of the clearance when they are co-administration with statins. The other clean farm consideration for biologics is renal and hepatic impairment. Generally, we don't need to plan for a systematic studies to investigate the impact of renal hepatic impairment because this is not really related to the metabolism or the elimination of the MAB. However, there are always some exceptions. Here you need to be careful about this. So if, for example, you identify any mechanism that can modify the target due to the renal or the hepatic impairment, so you may consider that there is maybe a modification of their clearance if you have, for example, a TMDD profile. In terms of renal excretion, we know that the cutoff for glomerular filtration is relatively low, between 50 or to 69 kilodalton according to the guidance. However, for molecular antibodies, probably this is not relevant because the molecular weight is much higher. However, if you are investigating a biologic drug with a molecular weight lower than these cutoffs, then you need to consider the investigation of the renal impairment. In any case, during your development, you need to collect the relevant information and include this data in the population PK analysis to see if there are any trend or significant covariate in terms of renal or hepatic impairment. The other clean farm consideration is about the cardiac safety and the QT prolongation. Also, this is generally not needed. We don't need to plan for a formal TQT study. Because we consider that the monoclonal antibodies are too large to cross the plasma membrane, hence they are enabled to block the ion channel. However, there are some rare examples of MABs that are able to prolong the QT through other indirect mechanisms. For example, this was described for denosumab, possibly through the disruption of calcium level. So in this case, you don't need to conduct specific study. However, the authorities will ask you to collect the data. Generally, you collect concentrations and matching ECG data and conduct exploratory or graphical analysis to investigate and to identify if there are any relationship between the prolongation of the QT and the concentration of your drug. Now to conclude this presentation, here you have a general clinical pharmacology plan for biologics. So as usual, you need to collect pharmacokinetics and pharmacodynamic data through all the development from the early stages to the late stages. The immunogenicity assessment is also important at all the stages of the development. Concerning the cardiac safety, you need to collect PK and matching ECG data to conduct this exploratory CQT analysis. It is better to collect this data at the early stages to facilitate the development and to limit the cost of this investigation and to exclude any risk from the beginning, and don't wait the late stages to explore this. The manufacturing changes are part of the development of the drug, so you need to establish a bridging strategy, and generally this is more critical at the late stage of the development. So if you have any change occurring during your pivotal studies or just before the BLA, so you need to consider a specific comparability clinical study. Population PK and exposure response analysis are also usual investigation for the development of biologics, and these are useful at all the stages of the development to select the dose and explore the exposure response relationship. The pediatric strategy was not addressed in this presentation, but also very important depending on the disease and the importance of this treatment for the pediatric population. So specific studies may be planned for the biologics drugs. Then you have some not mandatory, but studies which could be recommended according to the drug profile. This is the case for drug-drug interactions. If you have identified any specific mechanism relevant to induce an interaction between drugs. And for the renal hepatic impairment, of course, would depend on the drug profile. Otherwise, you need to include this investigation in a regular population PK or PK analysis. So this slide concludes this presentation. So thank you for your attention, and I'm happy to answer any question, if any. Thank you.
All right, thank you so much, Khalid. We've got a lot of questions from the audience, which is fantastic to see. Someone would like to know, for monoclonal antibodies, how sensitive is their clearance to charge or changes in the isoelectric point? And do you have, what would you say is the ideal isoelectric point?
Very interesting question. I think this question is relevant really for the early stages of the development. So this will depend on the, I would say, the target product profile. So depending on the targeted, for example, half-life, etc., you may modulate this isoelectric point to target specific half-life. I don't have any example in mind to give you a precise information about what could be the ideal one. But in my slide, you can see that there are some examples on which direction you will modify by increasing or decreasing the PI to have a rapid clearance or slower clearance.
Thank you. Someone else wants to know, is there a predictive tool that considers charge and the polar surface area of monoclonal antibodies in addition to the HFCRN binding to predict the half-life?
Also very interesting question and relevant to the early stages of the development. To be honest, I'm more specialized in late stage development, so I'm not really maybe the good person to answer this, but I can take this question and send you the information. Probably my colleagues from SimSip with the PBPK modeling and simulation tools have all the tools necessary for such predictions.
Fantastic. Do you feel that you need to conduct a PK bridging study, even if the formulation is identical with the pre-filled syringe? So I guess saying the formulation between a vial and the pre-filled syringe. Are a human factor study and a CMC in vitro comparability study, are those adequate?
Yeah, very excellent question. We may thought that the formulation, if the formulation is the same, we don't need this bridging as for the small compound. However, we know that if you change the device, you may introduce additional variability factors. So there's one known example about the difference between the devices is the immunogenicity. We know that, for example, depending on the manufacturing process or the presence of some particles in these devices, the immunogenicity profile may be different, and this is really critical in your development. So we know that the immunogenicity may impact the PK profile. So if you are increasing the incidence of the neutralizing ADA, some subjects may negatively be impacted by this by losing the efficacy. So you need to know it when you are switching these devices. And in addition to the immunogenicity, it will be around the dose precision. So the human factors, of course, can address how the subjects can use the device. Are they able to administer it correctly, etc. So there is really a difference, for example, in old subject with rheumatoid arthritis impacting the fingers where they cannot use it easily. However, in the human factor studies, you don't have the information about the PK. So for this reason, you can see more and more sponsors collecting the PK data in a real world setting to have a precise information about what is the impact of these human factors on the PK.
Great. So, question about trial design. So crossover studies are the standard in small molecule drug development. Why are you saying that a parallel design is a preferred study design for monoclonal antibodies?
Very good question. Unfortunately, for the specificity of the monoclonal antibodies, we know that their half-life is ranging between 11 to 13 days, depending on the product. So theoretically, you can conduct a crossover study, but this will take you maybe a year or 2 years to complete your study because you will wait for a long time to have a total washout. And even if you wait for a total washout, you may have also the immunogenicity, which will be not present in the first sequence and will impact the second sequence of administration. So for this reason, generally with the biologic drugs or monoclonal antibodies, we go directly for a parallel design, even if the number of subjects is relatively high.
Regarding drug-drug interactions, if there's already a product on the market for an anti-IL-23 antibody and has already tested the potential effect of this antibody on CYPs, do I have to do this DDI study again with my antibody if my product also targets IL-23, or can I ask the regulatory authorities for a waiver?
In my opinion, you can defend the waiver. Of course, you need to present all the data you have with the authorities to validate this. So this will depend also, I think, on the disease. If you are targeting the same population and the disease in terms of severity, because the modulation of the cytokines will depend also on the disease severity. So maybe the waiver will be granted. However, if you are developing the same, for example, on anti-IL-23, but in different population or in different segment of the disease severity, probably they will not grant a waiver. They will ask you to generate new data, because for the moment we still in the early stages of the exploration of this effect, and we don't have the answers or to all the interactions. And we know that in terms, for example, in induction, modulation of the expression, this is quite complex mechanism, and simple studies may not be able to answer to all the needs in terms of DDI.
Great. Do you feel that a concentration QTC analysis has to be done?
Yes, now it's really mandatory, even if the authorities are now clear about the waiver for the TQT studies. So this is, I would say, relatively a closed topic. You don't need to ask each time a waiver for a TQT study. However, they will always request to have this security analysis. So this is relatively simple analysis to conduct. However, what is complex is to collect a clean data, meaning that you have the PK with the matching ECGs, and these ECGs need to be conducted in a robust way, for example, in triplicate ECGs to count for the variability, etc. For this reason, I was saying during the presentation that it is preferable to conduct this in the early stages to control this variability and to avoid any, I would say, false positive signal.
Great. This looks to be the last question from our audience. I think the audience has asked a whole bunch of really fantastic questions. Someone would like to know, Khaled, when you would recommend using pharmacometrics or model-informed drug development within the drug development phases.
The MIDD is, I think, really informative, specifically for the drugs where you have identified a clear exposure response, or you have specific questions where the modeling and simulation could be helpful in your development. I would recommend to have this MIDD consultation quite early. As soon as you have the first data establishing this, for example, a PK-PD relationship, and you develop the first model, I think it's very helpful to go for the MIDD consultation. Yeah.
Fantastic. Looks like we've got one more question. Someone would like to know what are the reasons for performing population PK analysis in monoclonal drug development, prediction in special populations, or are there any other reasons?
The population PK analysis is generally conducted in a standard way as we conduct it for small compounds. So the first objective, of course, is to evaluate the sources of the variability and the relevant covariate. So it is very important to investigate this in the population. Generally, we confirm the effect of body weight, which is really specific for monoclonal antibodies, but sometimes we can identify additional variability factors, very important, such as the immunogenicity, with when you have, for example, a subcategory of subject developing immunogenicity with neutralizing ADA. So this subpopulation will have a different clearance. It's also important to have this variability factor to conduct a prediction and simulation in either special population. For example, this is rare, but you may have this renal hepatic impairment impact. However, it's often used also for the pediatric development. This is very helpful. We know that the body weight effect is relatively straightforward in monoclonal antibodies, so it's quite easy to develop models taking into account this with the allometry to extrapolate and define doses for the pediatric population using a POP-PK approach.
Fantastic. Thank you so much, Khaled, and thank you so much to our audience for their attention and their fantastic questions. Can you go to the next slide, please? Just a few short announcements before we wrap up. We have an upcoming event that you will not want to miss. In October, we will be having a free upcoming virtual symposium called the New Horizons in Pediatric Drug Development, which will take place online on October 28th and 29th. We've got an incredible lineup of speakers from FDA and industry and academia. You don't want to miss it. And you can register for this event at www.newhorizonpdd.org. On behalf of Certara, I'd like to thank you for attending this presentation. This concludes the webinar. Goodbye and have a great day.